Sanger sequencing chain termination method. Here is a DNA sequence to be analysed. It's a single strand of DNA. In our conical flasks, we have the single-stranded DNA to be analysed. We have primers to start off the replication and DNA polymerase enzyme and the normal nucleotides, so deoxyguanosine triphosphate and the cytosine, adenine and thymine versions. If we look at the structure here of the deoxyguanosine triphosphate on the left, you can see you've got the OH group of the deoxyribose sugar. What happens in this Sanger sequencing method is we also use these dideoxy nucleotides. So here's dideoxyguanosine triphosphate. If you notice, it has no OH group at the bottom there of the molecule. If the dideoxyguanosine triphosphate is added onto the new DNA chain, then it actually terminates the chain because it has no OH group on there. So we have 4 conical flasks. Each will contain what we have mentioned above, but in addition, we're going to add to each of them a different dideoxy nucleotide. So for example, in the first one, we've got dideoxyguanosine triphosphate has been added into the primers and the DNA polymerase and the normal nucleotides. In addition, a different fluorescence coloured dye has been added to each of the molecules of the dideoxy nucleotides. So let's look what happens with the dideoxyguanosine triphosphate. So if we look at what could happen with the DNA when it's been replicated here, the top strand here is the original strand, and then we've got the complementary strand by inserting new nucleotides. So here we've got C, cytosine, thymine, cytosine and guanine. So if the normal guanine gets added in the first time, it will carry on copying onwards from there, and you'll get the A, T, T, A and G added in. And if it's the dideoxyguanosine triphosphate, then that will terminate the strand at that point. So we've looked at this first example, and the one at the bottom shows you what would happen is if the first guanosine that was inserted was the dideoxyguanosine triphosphate, then that will terminate the strand really early. And here's what could happen with the dideoxycytosine triphosphate. So if that is the first thing that is inserted in on the complementary strand, then you're going to terminate the replication really early and have a really short strand. That's the bottom example here in the diagram. Or if you have the normal cytosine first, then a thymine, and then the ddCTP, you get this medium-sized strand, or it might get right past that and create this longer strand. So there's the 3 possible results we can get from this example. Here's what could happen with the ddATP, the 2 results that you could get for that. And here's what could happen with the ddTTP. So again, there's 3 different results there as well. So here's all the possible results of what could be produced depending on where the chain is terminated. The way that the DNA sequencing is done is that gel electrophoresis is carried out. If we think that the DNA fragments are moving from the top of the diagram here down to the bottom, then the smallest fragments will move the fastest and furthest through the gel. So if we read it from the bottom, we've got the C1 has reached the bottom, so that would tie in with our smallest fragment. And then a T, and then a C, and then a G. So you can see that it's possible to work out the sequence of the original strand of DNA from this gel method. It can even be done in a machine where the analysis is carried out and could end up with a result like this, with the smallest particles moving furthest through to the right, and get the DNA sequence that way. So I hope that has helped you to understand the Sanger sequencing chain termination method for sequencing DNA.